Welcome back to another video. Dear students, in this video we'll talk about meiosis 2, but for a short recall, I just want to tell you that in the previous video we talked about meiosis, and you learned meiosis is basically a reduction division and it is happening in the germ cells, and this is producing four daughter cells. Like one parent cell is dividing itself into four daughter cells. This is happening in a way that the diploid parent cell is producing four haploid daughter cells. And the haploid means half of the number of chromosomes as compared to the parent cell. Like if, for example, the parent cell is having 46 chromosomes, then they are reduced to 23 in each daughter cell at the end of the meiosis. We'll continue with meiosis 2 here. I have just mentioned all the phases here. You can go through this description. Okay, we'll see a short summary of meiosis 1. What happened in meiosis 1? Meiosis 1, in this, the parent cell was entering in Cytokinesis 1 that was having four phases. Prophase 1, in which we had 5 sub-stages like that leptotene, zygotene, pachytene, diplotene, and dikinesis. After that, metaphase 1, then anaphase 1, telophase 1, and cytokinesis 1. Now you see here, these cells, they will enter the next phase of meiosis, which is meiosis 2. You can see, dear students, here that the chromosomes, they are dissolved here. Okay, but once the cell enters meiosis 2, then its first phase is prophase 2. So here you can see this chromosome, like this chromatin material, it will start condensing itself and the chromosomes will appear like this. Okay, at the same time, the nucleolus will disappear and a nuclear envelope will start disappearing. The centrioles will move towards the opposite pole and they will form this spindle fiber. Why is this important? Because a spindle fiber will attach themselves to the chromosomes, okay? And the point to which they are attached, that is called centromere. So after prophase 2, this cell will enter the metaphase 2, in which these spindle fibers will arrange these chromosomes in the center at the equatorial plate. Here you can see. After this, once the chromosomes have aligned in the center, these cells will enter anaphase 2, in which what will happen? Now you can see here these sister chromatids, they were attached with the help of centromere. Now in anaphase 2, the centromere will split. Once it splits, these sister chromatids, they will also separate and they will move with the help of spindle towards opposite poles, and at the same time their length will reduce. You can see in both of these cells. After this, once these sister chromatids, they will separate and they will start moving towards opposite poles, the cell will enter the telophase 2. Here you can see easily that these chromatids that will now be acting as the independent chromosomes, they will like their movement will cease, their movement will stop, and they will act as the individual chromosomes here, like the independent chromosomes, and at the same time the nuclear envelope will reform, the nucleolus will appear, all the other membrane bound organelles like endoplasmic reticulum, Golgi apparatus, everything will form here in telophase 2, and you are having the centriole at the terminal. Okay, after this telophase 2, the cell will enter cytokinesis, because you can see here the nuclear division has been completed. Still, we are not having the proper divided daughter cells. Okay, so the cytoplasm will actually separate in cytokinesis 2 after forming this cleavage. The cytokinesis, in the cytokinesis, the cytoplasm will be divided, and in the end we will have four haploid daughter cells. So this is how you see how this parent cell is divided into four haploid daughter cells. This is the end of meiosis. I hope that you understood it quite well. Now everything has got its importance. We'll see the significance of meiosis division in us and in every species. This meiosis is happening in one way or other. So why is it important? It is basically maintaining the constant number of chromosomes. As you saw that in the end we got the haploid daughter cells. This is basically happening in our germ cells. So our gametes, they are haploid in number, like they are having the half number of chromosomes. Like in us we are having 23 chromosomes in our sperm and in our egg cell. So why is it important? It is important because once the sexual reproduction is happening, then these germ cells, sperm and ovum, they will form zygote after fertilization. So this is how they are maintaining the constant number of chromosomes, like 23 from the egg cell and 23 from the sperm. They will fuse together and they will give you 46 chromosomes in the next like in your offspring. So this is how we are maintaining our constant number of chromosomes just because of meiosis. Besides this is also important for the genetic variation. As you saw that in crossing over, there was the exchange of segments taking place. So this is very important for variation. Okay, we are not the exact copies of our parents. We are having certain variation within us, and this is just possible because of meiosis. And how is it possible? Just because of the crossing over that is happening in prophase 1. So this variation is basically important for the evolutionary process of every species. I hope that you understood the significance. Now we'll see meiotic errors. There are so many meiotic errors happening, like if the homologous chromosomes are unable to separate from each other, then this process is called non-disjunction. Okay? And if such cell fuses with the normal cell, then it will produce the abnormal cell. This condition in which instead of 46 chromosomes, if we are having 47 chromosomes just because of this non-disjunction, then this is called trisomy. Okay, and this is very threatening to the offspring that is just coming. So it is totally destructive. I hope that you understood it. Okay, and if you have any question, you can ask in the comments. I'll come with the next topic very soon. Till then, take care.